Thank you all for joining us. We're going through some of the works that have recently become part of the Envisioning Intelligences exhibit, which is the third decade of the Places and Spaces exhibit. And our first decade focused on maps of science, our second, interactive data visualizations, and our third, which we are forming, and this 24 hours is very much part of the forming of it too, will be on envisioning intelligences, biological and technological, all different kinds of forms of intelligence. And so what we're doing is looking at some of the works from this year's submissions. And after this session, we will look at the emotional echo of cultural intelligence, and we'll talk to the team behind that. And then at 5 a.m., we will look at responsibility, visualizing AI for Google DeepMind. And these are all really, really great works. And so we're very pleased to have the teams behind them here to talk to us and give us a sort of behind the scenes look at these works. And so I'd like to just introduce Liuhuaying. Is a data visualization specialist and faculty member at the Complexity Science Hub in Vienna. She creates interactive visualizations that transform complex scientific research and societal data into engaging, accessible experiences. Her expertise spans design and front-end development for interactive data projects at the interface of academic research and real-world applications. She has worked with organizations including the Massachusetts Bay Transportation Authority, MIT Sensible City Lab, and Smart Future Mobility in Singapore, and SBH Liuhuaying, producing interactive data journalism and research visualizations. Her work has received first prize in the 2019 TRB Innovations in Transit Performance Measurement Challenge, first place in the interactive category of the 2023 World Data Viz Prize, 2 gold awards in the 2024 Information is Beautiful Awards, one in the science, technology, and health category, and one as impressive individual, and most recently the City of Vienna's Promotion Prize in STEM for her contributions to making complex science accessible and understandable. And her work is returning to the Places and Spaces exhibit. Remember that her work was part of last year's, so the 2024 version of last year of the macroscope. So welcome. And also I'd like to welcome Amélie Desvars-Larrive. She's a faculty member of the Complexity Science Hub. She graduated in 2004 as a doctor in veterinary medicine from the National Veterinary School of Toulouse in France, with a specialization in tropical diseases of livestock and epidemiology. Amélie worked for 8 years in the Indian Ocean as a field epidemiologist and veterinary research engineer dealing with livestock and wildlife, especially rodents and bats. In 2013, she received a PhD in epidemiology from the University of Saint-Denis, La Réunion. Amélie did her first postdoc in the Department of Mathematics and Mathematical Statistics at Umeå University in Sweden, where her research aimed to better understand the epidemiology and ecology of the zoonotic disease tularemia. Her second postdoc was at the University of Veterinary Medicine, Vienna, in conservation medicine. Her project focused on urban brown rats and their role in the epidemiology of emerging zoonotic diseases as a source of antimicrobial resistance in the city. Her research interests include infectious disease epidemiology, including zoonotic and emerging diseases, veterinary epidemiology and public health, modeling of infectious diseases, the One Health approach, and rat-borne diseases. Amélie is also an associate professor in infection epidemiology at the University of Veterinary Medicine in Vienna. Welcome. Thank you so much for coming and presenting today. And with that, I'll just give the floor to you.
Oh, hello. I'm happy here today with Amélie to talk about or share how we created the zoonotic web visualizations. As Todd mentioned, that this piece is curated by the Places and Spaces exhibit, and also has won this gold award last year in science and technology and health category. And now it's also displayed at the Vienna Museum as the modern contribution to knowledge for all, this isotype exhibition that marks 80th anniversary of Otto Nora's death. If you're in Vienna, you can visit there until April 5th. And even though this is aimed at the public, this visualization, and the visualization actually is grounded in scientific research led by Amélie, and is also a part of the paper published in Nature Communications. And you can also see there's also other researchers behind this piece. It's not a single person's work, it's a collaboration work. And this kind of a collaboration is also quite typical at the Complexity Science Hub, where we have over 80 researchers working on around 14 topics in complex systems. And how this works with visualization, that our scientists work on their research, and then once they have results, they will come to our visual team, which is led by me. And then we will discuss what visualization to create. And then once we have it, we will talk to our communication team, and they will pack the paper and the visuals and together and disseminate it to the world. And our audience isn't just academic peers, but also general public, like policymakers and even school kids. And you can find all our visualizations online on this website. And from my experience working on those visualizations, creating the good ones for science communication really starts with understanding the research and the methods behind it, not just the data set itself. So once we understand that, we can tell, okay, what to show, what to tell, and how to show those results. And that's why today we are lucky to have Amélie here to share more those research behind this project. So welcome, Amélie.
Yeah, thank you very much. So I will not talk too much about what I do because it's been presented just before. Just to mention that I'm very interested in interfaces. That is to say, where disease would jump between species. And that's what we have explored here in the zoonotic web. And what I've also been experiencing in the past year that animal health data are usually very scattered and unstructured. And we've been very interested in the past year to make those data readily available for analysis. And before I start to describe you the work we've done together with Liu, I need to explain you what is One Health. So One Health is an integrated and unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystem. And very importantly, this approach recognizes that the health of humans, animals, plants, and the environment they share are closely linked and interdependent. And to illustrate a bit the need for One Health approach, I would like to show you this picture that describes zoonotic systems. So in real world, animals live in certain places. They may share the environment with humans, with plants, and we also have water. Some pathogens are circulating in the human population. Some pathogens are circulating in animals' population, sometimes across a species, and many of them actually can jump into humans. 60% of human disease are originating from animals. So the influence of the environment is also very important on this pathogen survival. So we can see here that we have a very complex system with multi-agent. And to study this system, to fully grasp the interconnection between the various components of this system, of the zoonotic system, we need to adopt system thinking. So system thinking is a way of making sense of the complexity of the world by looking at it in terms of walls and relationships rather than splitting it into its part. So conceptually, maybe you have already understood where I would like to go. The One Health approach is very interesting for us because it really grasps conceptually the interconnection between the health of animal, human, and the environment. And technically, network analysis is also a very attractive method because it's quantitative and reproducible. So we can put number on these interactions. So we were interested into zoonotic interaction in Austria last year. And when we started thinking of it, we realized that there were no data sets of zoonotic agent that could have been found in Austria. And not only in Austria, but no country has a data set that would be readily available to look at their zoonotic interactions. Global data set exist, but they are too coarse to really understand local context because you understand that the environment is very important and it also influences very much as interactions. So what we did, we've collected our own data to build our own data set. So we've screened the literature using a very systematic method. It's called PRISMA guideline. We've collected more than 200 publications. We screened them using very specific criteria, and we ended up with 246 papers. Those some were scientific articles, some were national surveillance reports, some were theses. And we've collected these publication from 1975 to 2022. We've extracted data from all this publication and we've extracted data manually. So this was a tremendous work because we really wanted to understand which sources have been infected by which zoonotic agent. And not only we wanted to have this interaction, we wanted also to know how it's been evidenced, whether it by serology, by molecular evidence, by culture. So we've collected all this data, and for each interaction, we've collected many epidemiological data, ending with 48 fields, including taxonomy that we have all harmonized between species and agent. And we have created a network of zoonotic interactions. So the network is undirected and it represents actors that interact, so sources that could be animals, arthropod vector, environment, or food, and zoonotic agents, so almost 200 of each of them. Zoonotic agent included bacteria, virus, and eukaryotes. By eukaryotes, I mean, for example, fungi, protozoan, or helminths, for example. And we have that different category of sources, so where zoonotic agents have been found. Of course, human, because all include zoonotic agents, so that could infect human. Different kind of pets species, livestock species, wildlife. So arthropod vectors include, for example, ticks or mosquitoes. Different kind of environment like water or soil, but also public lavatory, for example, and of course, food. And when we found in the literature that one infectious agent has been identified in a source, we put in the network a direct link between them, meaning that this agent was identified in the source. So for the vector, the food, and the environment, all included direct evidence, so either molecular identification or culture. And for human or animals, it could also have been serological evidence, meaning presence of antibodies. And then we introduce a concept of a zoonotic web, like a food web if you want, but instead of energy flow, the network shows infection or contamination. And this is the figure 3 of the paper. So as you can see here, the zoonotic web is a bipartite network. That means a graph that contains two types of nodes, the zoonotic sources that are represented here in colors, and the zoonotic agents that are here in black. And they are connected by edges which represent infection or contamination. On the graph, you see different size for the nodes and they represent the degree centrality, which means the number of links a node has. And in the epidemiological context, the node degree centrality for each zoonotic source corresponds to the zoonotic agent richness. It means the number of taxa directly or indirectly, if we speak about antibodies, detected in the source. And similarly, the node degree centrality for the zoonotic agent corresponds to the zoonotic source range. That is the number of sources from which it's been directly or indirectly evident. So we would call it host plasticity, and we adapted this term to source plasticity for this work. And we also had in this network what we call potential transmission chain that we represented by dashed lines. So to better illustrate what are potential transmission chain, I will use the rat subsystem. So if we look at rats, we found in the literature that different zoonotic agents have been evidenced in rats, so Cryptosporidium, for example, or Leptospira or Giardia. But in other publications, we found that these pathogens have also been evidenced in wild boar or in the common wall or in cattle. So even if the transmission chain from a rat to cattle has not been evidenced, for example, for Leptospira, our work allows us to evidence potential transmission chain because instead of looking at individual study, we look at them together. And to study the cross-source transmission and rank the sources by their importance in the network of zoonotic interaction, what we did from the bipartite network, we projected it into a one-mode network of source-source interaction, where each node would represent the sources. And the ages between the sources would represent exchange of pathogens between two sources, and we've weighted the age by the number of zoonotic agents shared between two sources that we still adjusted by research effort. So we wanted to study the importance of the source in the zoonotic system, and for that we used 4-degree centrality. So the centrality matrix, sorry. So the degree centrality, which is for a source, the number of other sources it's sharing pathogens with. We use also the strength centrality, which is the sum of the zoonotic agent shared by a source. We use betweenness, which identifies nodes that could act as bridges between different source community, and closeness, which is a distance, the average distance to all of the nodes, and it represents how close a source is from other sources. So to summarize a bit, degree and strength centrality would represent direct interactions between sources, while betweenness and closeness would represent indirect interactions. And we've ranked the sources by importance using this 4 centrality matrix. So this is a table showing the top 10 ranking by centrality matrix. To summarize, what we found is that human, chicken, cattle, and meat are the most influential node in the network, which means that the node that shares the highest amount of zoonotic agent. And interestingly, we also found that wild boar, for example, or dogs or cats, they are, or Ixodes, the tick Ixodes, rank high by betweenness centrality, meaning that they could link two communities that would be separated otherwise. Here is a figure 5 of the paper, and it shows communities in the network of zoonotic interaction. So we were interested in looking at clusters of zoonotic sources that would share similar agents. So we used the Leiden algorithm to find them, and the figure shows a different community and the agent shared within this community. For example, if you look at this graph, you see the number 1. Here is a community 1. And this community 1 primarily comprised central hosts, so those having a high value by centrality in the network, and those are species very close to humans. Livestock species, pet species, synanthropic species like rats, for example, or game species, meaning that the risk of spillover to human, of transmission of endemic zoonotic to human, mostly come from the species that are the closest to us. Another example of community, for example, if you look at West Nile virus, this is a community 5. Here we mostly have bird species, but also horse that could be infected by West Nile virus, and the mosquito culex, which is a vector of West Nile virus. So that's it for the paper and the scientific part, and I leave the floor to Liu to continue with the vid. Thank you.
So you have seen so far charts, visuals, and you might notice that that's why we chose network visualization. That's why, because it's based on scientific results, and why we present it certain ways. This is why, because we have network analysis, we have community detection, those methods we need to present. And then the question here might be that how we translate those into an interactive version instead of the static ones. And apparently you can see this, the interactive one network is similar to the one in the paper, the figure. And actually I first created this static one for the paper and then turning it into this interactive piece. And one noticeable change is that I removed the labels on the figures and I used the icons for the nodes instead. And then labels only appear when you hover over nodes here. And then it as we mentioned before, like there are like almost 400 nodes. So it would be too much work and also unnecessary to create individual icons for every node. And our eyes cannot distinguish those tiny details anyway. So identifying which icon were actually needed, it was a big task for me. And there might be automatic tools for this, but what I did was go through all the nodes manually and create a mapping system for myself. And take the host nodes, for example, here we have like 155 nodes for hosts. And each with the scientific names and family names, order names, you can see it in this table. And for different type of birds, for example, here we see like blackbirds or sparrows, I used a single bird icon at class level, avis, because they are all avis. But for example, for parrots, then I used a different icon because people are more familiar with them as a pets. So they invoke a very different image for people. So they get their own icon at this family level, Cetatidae. Use this kind of logic, and I end up creating only 25 icons for 155 node hosts. And on the right side, you can see this is my sketch notes for all the icons that I need to create for them. For example, you can see I used one icon for both ducks and geese, and also one icon for tortoises and turtles, simply because people cannot really tell the differences when you see a tiny icon. But also it's tricky. For example, for creating the icon for ducks and geese, I need to create an icon that has to be generic enough to represent both. So this icon cannot be too duck and cannot be too goose. And there were also some species, might notice that those, even a common English name, I wasn't familiar with. So I marked those in Chinese. So I use Google Translate and I use Google search to see what are those animals and how they look like to create those icons. Another, it's kind of straightforward for vectors though. I decide that all ticks share one icon, or flies one, and all fleas another, because people usually cannot tell the tiny difference, how many legs, for example, for the insects. So, and then use this approach, I created a total of 45 icons. And you can see I actually also had a struggle with sausage, meat, and fish here. Technically, fish and sausage are also meat, but in everyday life, we don't group them the same way. So for example, you don't find fish and sausage in the same section of the supermarket. So I separated them into different icons. And those icons, of course, appeared throughout all versions of visualization on the website, including community visualizations. And compared to the one in the paper on the right side, I kept the node link structure and added this position transition to show how those communities come from. And that's why I also kept the original links instead of using the edge read for degree here. I also kept the source nodes, make them visible instead of showing only agents in a figure. And another very important component for this project is the scrolling telling, where user can scroll down and to read more and to see what they should learn. And this technique allows us to explain all the information Amélie just introduced, like what the links mean and how those potential transmission might happen, and also allows us to highlight insights from network analysis, like degree centrality and bridge nodes and important nodes extra. And of course, to support the audiences without the scientific or diseases backgrounds. And we added more fundamental explanations. There's also things that I learned myself during this project. For example, quick explanation of differences between bacteria, viruses, and eukaryotes, and some familiar example of each. And when you look at those data for these agents, and you can actually see they have their own structural hierarchy, like superkino, order, family, extra, is a complicated structure here. And so I actually plotted them first in an online tool called row graphs. And then you can clearly see there's a 4 level of hierarchies of those a group of agents. And based on this, I designed and created this tree map to represent the structure visually here. And from there, through this storytelling, we introduce each of those branches so the audience can learn what they are seeing here. And see here, we can introduce examples, we can introduce the structures, and also the same techniques, then we can introduce our sources in groups. And then why we added some content, right, to help the general audience, and we also actually left out certain results from the paper. So there's a coming, if you look at paper, if I don't know if you have to look at paper, and then of course, besides there's 2 main visualizations here, you will see more results actually here. It's not included in the visualization, the interactive pieces. And this brings me to my next question to Amélie. What else can we find in the paper?
So when collecting data, we also have collected data on, was this pathogen imported? Was this pathogen discovered for the first time in Austria? One very important notion in epidemiology, and you've all heard about it during COVID-19, is the notion of emerging infectious diseases. So we were interested in looking at those zoonotic pathogens that were evident for the first time in Austria. And what we discovered collecting the data is that over the 50 years of study, 8 zoonotic agents were completely new to the country. So we often have this idea that emergence occurs maybe in Asia or in this biodiversity hotspot, but actually also Europe have emergence. And here we mean emergence by agents that were not present in Austria. They were present in other parts of the world, but have been introduced in Austria, and all of them in the past 20 years. For example, Usutu virus in 2001 has been evidenced in Austria, and that was the first discovery outside Africa. So that was very interesting for us to look at that. And overall, it means 1 emergence every 6 years over the study period. All of them in the past 20 years, which means the frequency is increasing. And 4 of them are actually vector-borne diseases. And we all know that vectors, mosquitoes, for example, or ticks, are highly influenced by climate change, climate warming. So we can have, we have to keep in mind that that emergence might occur more frequently in the future. And as I mentioned it before, we are very interested in this One Health concept. So we looked at very specific structures in the network. We look at 3 clicks, which are triangles of nodes. And the 3 clicks, we only look at those 3 clicks, including a human node, an animal node, and environment node. So we have this One Health triad and One Health interface, where we think usually in the literature that where spillover would occur. And we classified there's One Health 3 clicks we ranked them using the number of pathogen exchanged at their interface, summing the weight of the edges at for their 3 clicks. And if you look at the left figures, you can see that the distribution of the number of pathogen exchanged at this interface is right skewed, with most of the interface showing less than 20 pathogens, zoonotic pathogen, but some of them on the right side showing more than 40 zoonotic agents. And if you look at the 6 top ranked One Health interfaces, One Health 3 clicks on the right, you can see that cattle and food products are highly involved in zoonotic contamination to humans. So what's next? You may have noticed that this zoonotic, the network of zoonotic interactions, the zoonotic web is quite static and it's not directed. And of course, we all know that zoonotic agent transmission is not symmetrical. So some agents are only transmitted from one species to another one and not bidirectionally, or more often in one direction than the other direction. So we would like now to study the directionality of the link. We are also expanding the zoonotic web to Brazil and France, and we really would like to expand it globally. But you might think, yeah, manual data extraction would be very much too much work. So we would like to use some maybe LLM or automated data extraction. So we're working on that to have more data to expand the analysis and go maybe also deeper in the method, maybe use multiplex, because beyond a static visualization or analysis, what we would like to do, still thinking of this One Health concept, is looking at the influence of the social ecological system on the zoonotic interactions. Yeah, that's it from my side.
Yeah, this is from our side.
Thank you so much. This has been super informative. When you make these visualizations, well, I guess this project as a whole, you mentioned that these visualizations find their way into classrooms and are used by various different audiences. Do you engage in sort of user evaluation or anything like that to sort of kind of check whether what you're trying to get across is coming across in the way that you intend?
So I use it in the classroom in the veterinary school with a student. So far, the feedback is they use it to, to learn mostly, right? It's a learning experience for them. So it's a very specific public. That's a vet student, obviously. I don't know if you have a feedback from the public in general, if they give a feedback to you. So I'll let you answer to that.
Yeah, so usually we do not have like systematic way to evaluate this. Mostly it is collect the feedbacks from people we know. Usually for me, there's 2 layers always. One layer is it has to be scientific correct, like scientific makes sense. And then that's why I often also use this, give this to other scientists here and say, does it make sense to you? Like, even they are not working on this field, but they may use the similar methods or they understand the science better than me, and they can tell the way we describe, because we usually don't want to use the so techno jungles or technical words there. But then if you phrase in other words, still makes sense in science. And we want to make sure that is correct. And then the another layer is we want to get the feedbacks from people, they do not have scientific training. And then we have our communication team, they are more like a journalist background, and they often also give us some feedbacks, say, okay, this is too technical or like too difficult to understand, or they didn't get what I mean, if this is this network. I mean, network usually is simpler, but some methods like modeling or aspects, and they wouldn't understand how it works. Or sometimes the data is estimated, it's not a real data. It's estimated results, and then they would like to know more about that. So we usually collect those feedbacks from two groups. Yeah.
What was the most challenging thing about this project? Was it putting the data together? Was it kind of figuring out the best way to present it? Was it obstacles in terms of bringing forward the One Health framework? What were some of the defining challenges of this project from both of your ends?
That. So obviously collecting the data has been a tremendous work. And then also we faces one issue, which was the taxonomy evolves. So the name of the bacteria from 1975 to 2022 has changed for some of them. So we had to harmonize. So we use the NCBI taxonomy database to harmonize all virus, bacterial, and eukaryote names. And the same for the animal host and vector species. So this has been also a lot of work to harmonize all that. And then building the network also, because what we have usually in my field in epidemiology is network of host pathogen interaction. So only animals and only pathogens, and sometimes only mammals and viruses, or sometimes only primates and bacteria. So it's very narrowed area. And because I was very interested in this One Health concept, I put everything, we put everything in the network. And this was very challenging to give a meaningful significance, some meanings to this analysis, to the network analysis. So that's also why we use simple metrics, node metrics, only 4 of them. Making sense of all this data together, it was quite new, and that was very challenging, I think. And in the end, it was quite successful. But yeah, I think that was the most challenging part. Yeah, maybe you have also some challenge, to share.
I mean, creating icon is a challenge for me. I report. And another challenge is network visualization. I mean, because for me, everything can be a network. Like you define nodes and you define relations, and then you can make a networks. But the point is how to make it meaningful and present it. And also when you involve the list metrics, like centrality, it's very abstract concept for people who are not doing network science. And you have to explain this well, and then how do you transform this information into something that people can understand from non-scientific background is also a challenging for me. And that's why I think this storytelling component, it's kind of essential here. Yeah.
Yeah, could you talk more about that? Because I love storytelling as a form, and I think it replicates sometimes the process of doing it. Like it feels like you're doing it along with the researchers as you're going, because you're getting all the terms, you're getting all that, everything is sort of set. It has this wonderful process to it. Is that also what attracts you to the storytelling of that in its simplicity, or what is it that you keep coming back to with this?
I mean, it's a technique that used a lot in journalism, right? Like news graphics, and that shows that there's a space that for you to tell more, and the engaging way to tell more, especially if you have a visual there. Of course, there's some cases that is not ideal to use storytelling, and if you don't have many transformation in visuals, for example, you don't have many highlights, then it's not a good techniques for this. And in this piece, it's very useful because it gives a lot of opportunity to highlight certain parts in a network. And we also can, because it's all about those nodes and the links. So it also allows me to transform the positions of this graph. And then people can understand, okay, now I'm looking different layout of the networks, but they are same nodes and links. I don't need to relearn everything again. And it's quite convenient. And also people get to know, okay, how this layout comes from this, and they come to this now. And then with some text, then you can you can explain what happened, and also have several slides to highlight certain layers. And of course, the tricky part is like how many slides you have to have. You cannot do too long, like half-hour reading. No, that's too much. So it's also very tricky for me to shorten some parts and to make it just more essential, and to show what is really need to show, what is not necessary. And some parts, like those links, and actually, originally we don't have that much explanations, but then we decide we have to emphasize, make this more clear, and then we expand the more slides there. Yeah.
What are Amélie, you mentioned using the One Health framework to in the future to maybe take the study broader, cover more territory, explore more methods. Are there other things that this One Health framework allows people to do, or other areas that it could help people illuminate in their particular fields?
Well, the One Health concept is widely used, so not only in infectious disease, but in health in general.
Yeah.
It's a way, of understanding that our impact of the world and the different global changes really influence our health. If the environment is not healthy, if it's polluted, of course, our health and the health of the animals will be affected. So this is a concept that is widely used. So in my field, in infectious disease, of course, if you have climate, global warming, if you have more rain, so the chance of infectious disease to occur more often and more intensively. That's the way how we use the One Health concept. But not only it's at the global scale, but it's also a very local influence on diseases. Some diseases are specifically occurring in very small region because the environment allows it. And also you have cultural factor. If you're some cultural or socioeconomic factor. If you're a hunter, you're more exposed to disease from a fox, for example, than anyone else, right? So that's what you eat. If you eat your meat raw, you would be more exposed to foodborne pathogens. So local practices are also very important in this One Health concept. And we usually say that it scales from very local to global. That's why we are interested in collecting data at national level. And I didn't show it here, but we also have regional data. So we can also disaggregate by region. So maybe at the scale of Austria, it's maybe interesting if we do, for example, Tyrol versus, non-mountainous region. But if you do it, for example, from Brazil, which has so different bioecological region, a bioclimatic region, then maybe it's very interesting to look at the local context and how zoonotic interaction shapes depending on the context. So that's how we want to use One Health here.
Fantastic. Thank you. Just one last question for you. What is the secret to a good collaboration?
Well, for me, I always think, of visualizing the work to transmit something, transfer knowledge. But working with Liu, I'd say it's amazing because, you come to her, I do a network in R, for example, and it doesn't look so good what I do. But then I show it to her and she's like grasping really the concept and what I mean. And then we talk a lot. And I would say now we've been working together since few years and we bridge really the language barrier that we could have at the first time, right? Being myself a vet and Liu being a visualization expert. I think now we really on the same page when we talk, and she grasps the meaning of the data and how we can communicate the data. So for example, this storytelling she did, I didn't know she would do that. And one day she sent me a link and it was all there, you know. She has worked on the concept, on everything. So I tweaked some words maybe, but it was already ready. So that's been amazing. So yeah, I think it's about also giving trust. I share the data with her, said, look, I have that. I don't know how to visualize it. What do you think? And she does something, and it's usually surprising. It's like, wow, or sometimes, oh, I don't like the color. For the community, I remember I didn't like the color. It was black at the beginning. And I said, oh, no, I would like a bit blue. And then she put some blue. So it's a lot of talking, exchanging, and yeah, meeting, I would say. Yeah.
Mm-hmm.
Yeah, for me actually is care. Like, I mean, I worked with a lot of scientists, and most of scientists are very focused on their own work, and they want to communicate more to their own peer, to be honest, which is good. They need to do this, or they talk more to policymakers, also good. But when we come to say, what about we communicate to more general audience? And then they don't know what that means. Like they don't know, then what should I say? Is this sentence technical or not? Or so they don't get a sense of what that means by communicating with general public. And then when I worked with Amélie, and then I said, okay, here, I think we need to explain more. I don't think people get it, understand it. And then she trying to understand and then also help me to correct those parts I actually misunderstood in the beginning. And then I feel like, oh, actually, I didn't fully understand the project maybe. And then we learn more. And then that's how we can end up this really amazing piece that for surely scientific correct, understandable. And then we're happy with this collaboration. So I think both sides need to care about this or group of audience or this. Yeah.
Mm-hmm. Well, and it is such a successful piece, and it's a testament to the ability of good collaboration. And I really appreciate you coming and talking about this and answering some questions and giving us an overview of this wonderful work. So thank you very much. Enjoy the rest of your Sunday, and thanks for giving a part of it to us today. I appreciate it.
Thank you for inviting us. Thank you being there.
Thank you very much. Bye.
Bye-bye.
Bye.